All right. Good morning, everybody. I am really excited to be sharing the research that I've been doing on TANGO2 Deficiency Disorder in a C. elegans worm model. All right. So off the bat, I have no financial disclosures or conflicts of interest to share with you. All right. So to start us off, I first want to talk about why am I studying a disease in people in teeny tiny microscopic worms? So these worms are actually a really interesting and very powerful model organism that have been used to study a variety of genetic diseases. So the behaviors that these worms exhibit are really well characterized and well understood. They have a short lifespan and reproduce quickly, so you can get a lot of little worms in a very short time span. So you can study you can get a lot of data in a short amount of time. Their genetics are really easy to work with. So the worms primarily reproduce by self-fertilizing. So you can track genetic mutations very easily. And they have very well-conserved metabolic pathways that are very, very similar to those that happen in people. And they require a lot of energy to maintain their daily worm activities. And for relevant to our group, these worms have these two genes that are very similar to TANGO2. The scientific term for that are gene homologs, and these two genes are called HRG9 and HRG10. They were first discovered by a group that was interested in this compound called heme. And so heme is a compound that's really important for oxygen transportation throughout the cell. So in humans, heme is a compound that's used in our red blood cells to help transport oxygen throughout our body. And so these researchers found that when they took heme out of the worm's diet, which is how worms get their heme, they found that this family of genes that's upregulated or turned on at much, much higher levels. So they're thinking that these genes are important for worms when they're trying to take in heme from their environment. And so looking at one gene, HRG9 in particular, they took some worms and they created a model where these worms were missing these two genes. So these genes didn't work the way that they were supposed to. And then they looked at some compounds that are similar to heme but act a little bit differently. So in one experiment, they fed worms this similar compound to heme called ZNMP that travels through the worm body like heme does, but it turns red. So it's fluorescent, and you can track where it's going in the cell. And so when they fed their worms this fluorescent heme compound, they found that in worms that don't have these TANGO2 lookalikes, HRG9 and HRG10, they were getting all of this beautiful red fluorescence inside the worm intestines. So these are two worm bodies, and these are all the nice bright red intestinal cells. And so that led them to think that maybe this heme compound is getting stuck. So the worms are having problems moving heme within the cell. They then tested this by feeding worms a slightly different compound called GAP. And this is a poisonous compound. So it looks like heme, and then in normal worms, they eat this. It gets into the cell, but the cell can't use it properly. And so because they don't have heme, the worms end up dying. And so they looked at the ability of normal wild-type worms and worms missing HRG9 and HRG10, and they looked at their ability to survive this poison. And so they found that when they fed these wild-type worms this poison, most of the worms died. This is what they expected. However, in worms that are missing HRG9 and HRG10, most of these worms were able to survive this, which is really unusual. So they came up with this model where they hypothesized that maybe TANGO2 equivalents are important in transporting heme throughout the cell. So normally, you have areas in the cell that have a lot of heme. They need to get taken out of there so that they can be used throughout the rest of the cell. And so maybe these HRG9 and HRG10s are helping to do that. And so when they're not working properly, heme is getting stuck elsewhere. So we were super interested in this line of thinking, because this is kind of different than what we had thought that TANGO2 might be doing. So we were able to get a hold of the worm model that this lab created, so they very generously shared that with us. And we took a look at some of the same experiments that they did. And we got some different results. So the first thing we tried is we tried that experiment with that beautiful red compound. And so N2 here these are normal wild type worms. So N2, N is for normal, you can think of it like that. And then we have our double knockout worms. So these are the ones that are missing those 2 genes that are similar to TANGO2 in worms. And we found the opposite of what they did. So we found that there's some level of fluorescence in the intestine. So imagine this is red. My pictures are in black and white. And we found that when worms didn't have TANGO2, the level of fluorescence in the intestine actually went down a little bit, which we thought was interesting. We weren't really sure why. But as we were observing the worms, we noticed that these worms were going through their food less quickly than our normal worms. So we thought maybe these worms aren't eating fast enough, and so that's why they're not getting as much of this fluorescent compound into their intestines, so their intestines aren't as red as normal worms. So to test this, we wanted to compare it to another strain of worms that we know has a problem eating. So they're missing a part of their throat that helps them sort of pump their bacteria into their stomachs. And so when we looked at the fluorescence in these worms, we found that these are EAT2 knockouts. And so we found that these worms have even less fluorescence in their intestine, which you can see down here. There's like almost nothing in here. So that led us to think that maybe some of what this other group had seen is less because of a primary heme problem, but is maybe more because these worms are just having trouble eating their food. So when we're thinking about the experiments that they did with this particular poison, we also took a look at those, and we also got slightly different results than they did. So across a variety of compounds, at low dose of this poison, we found that most of the worms were able to survive. And as you increase the dose of the poison, the worm's ability to survive this goes down, which makes sense. And then paralleling our idea that maybe our TANGO2 worms are able to survive this poison, not because they have any problem processing heme, but because they're just eating less of the poison. And so you can see this best at this 2 micromolar dose. So at this dose, about half of the N2s are able to survive. A little bit more of our double knockout worms are able to survive. And then our EAT2 worms that are really not eating much at all are almost all able to survive this poison dose. So that led us to think that maybe there's this other phenotype or behavioral profile that's happening in these worms that would explain what these other researchers saw and might have some interesting implications for us to look further into. So we did a little bit more work on that. We actually recently published a preprint, which if you want to hear anything about that, we have a link to that. And our wonderful lab tech also made a poster for us, so you can go check that out later at the poster session and hear more about what we did on that front. So then to move in a slightly different direction, this led us to do what could these TANGO2 equivalents be doing inside our worms? And so some data from some other awesome people who are going to be talking to you later has led us to think about vitamin B5. So we know from work that Dr. Satcher's group has done that in his fly model of TANGO2, when these flies are given vitamin B5, some symptoms similar to TANGO2 that these flies have tend to go away. So these are seizures. So when the flies don't have TANGO2, they have seizures, and then if they're given vitamin B5 at different doses, the seizures that these flies have go away. And we also know from some data from the natural history study by Dr. Miyake and Dr. Lalani that some of our patients that are taking these B complex vitamins don't get these metabolic crises, which is really exciting. So we wanted to learn more about what we think could be going on. Like, why is vitamin B5 able to help these flies and these kids? So I want to introduce, this is the idea that I'm kind of testing for my thesis work. So we know that vitamin B5 is taken into the cell when we eat it, and it goes through this process of synthesis, and it gets turned into this compound called coenzyme A, or CoA. And this is the only thing we know that vitamin B5 does inside the cell. And this CoA compound is really important for a variety of processes that happen within the cell. So about 10% of all cellular reactions, all of the chemistry that's happening in your cell, about 10% of that uses this compound in some capacity or another. So it's really important that our cells maintain a nice big pool of this compound so that it's able to be used in all of these different areas throughout the cell. And so this pool is maintained both by synthesis, using vitamin B5, and also by salvaging it. So once CoA is used in these reactions, it needs to get turned back over into vitamin B5 so you can keep making more of it. So because vitamin B5 is able to help TANGO2 patients, that makes me think that this pathway is probably okay. They can, our patients can still take up vitamin B5, they can eat it, and if it's helping them, it's probably getting turned into CoA. Which makes me think that maybe TANGO2 is acting on this side of things. So if you take away TANGO2 or take away AGG9 and AGG10 in my worms, maybe something's going wrong in this salvage pathway. So you're not able to maintain the amount of CoA that you need to do all of these different important reactions. And then when you supplement with B5, so you're taking all these vitamins that's giving the cells extra B5, you're able to make more of it through this synthesis pathway, and you're able to get this pool of CoA back up to a level that your cell can use, even though maybe something isn't quite working in your salvaging pathway. All right. So just to get into the chemistry just a little bit, you don't need to worry about all of the big words on this slide. The main point here is that vitamin B5, which is also called pantothenate, you might see me refer to that, gets taken up into the cell. It goes through this whole biochemical process, which is really cool, but I'm not going to get into the details of it. And it gets turned into coenzyme A, and then it needs to get recycled back into B5 or pantothenate. And so this is the molecular structure of CoA. I just want to draw your attention to this one intermediate that I'm interested in, which is called pantothene. And so this is this little segment of this compound. And the very last step of this recycling process is that this compound has to get split in half into, so you can get pantothenate back, which is our B5, and then it creates this other compound called cysteamine, which is used for other processes within the cell. And we're particularly interested in this guy right here because we've done some modeling of the TANGO2 protein using these in silico predictive pathways. So these are able to predict what we think the structure of TANGO2 might look like, which can tell us a lot about what we think TANGO2 might be able to do. And so in this structure, there is a predicted site where this compound could potentially interact with the TANGO2 protein. And this site also has a function where it could have the correct sequence that it needs to be able to cleave this compound back into pantothenate and create that other byproduct. So that's kind of really the biochemistry of what we think could be going on with TANGO2 in the cells. So the first big question to test is, do worms that don't have TANGO2 have a problem with their CoA pool? And so we worked with our wonderful collaborator, Dr. Roberta Leonardi, out of University of West Virginia. And so she's able to analyze the total amount of CoA in our worms using this complicated process called liquid chromatography, mass spectrometry. So she can look at all of these individual little compounds. And so we were able to find that when we have normal worms and our double knockout worms that are missing our TANGO2 homologs, when these worms are fed, so they're under normal conditions, they have an equivalent pool of CoA available. So the CoA levels in these worms are about the same at baseline. But if you stress the worms out and you deprive them of food for about 24 hours, and then you look at their CoA levels, we saw that these N2 worms, the normal worms, were able to more or less maintain this pool of CoA. However, the amount of CoA in the worms after they were fasted for 24 hours just plummets. Which is pretty indicative to us that this is a problem with maintaining this pool of CoA. So our worms may not be able to salvage CoA as efficiently as normal N2 worms. So if these worms have this problem, do these worms have symptoms of having a problem with CoA? And so we did some studies to look at the phenotype or the behavioral profile of our worms, and we found that these worms do have some differences from our regular worms. So we first looked at how long they're able to survive. So once they reach adulthood, worms have a pretty characteristic lifespan. They should be able to live 2 to 3 weeks after they reach adulthood. And so this red line is our normal N2 worms. This is how long they're surviving once they hit adulthood. And then this blue line is our double knockout model. So our worms missing TANGO2 homologs are not able to live as long as normal worms. We also looked at these worms' reproductive abilities. Not less for fertility questions, but because laying eggs for these worms is very, very metabolically demanding. It takes a lot of energy to make all these eggs from scratch. These worms are laying their entire body weight in eggs for several days in a row. So that takes a lot of energy for the worms to be able to do that. And so if these worms have a problem with cell metabolism or energy, they might not be able to reproduce as much as a normal worm. And so we were able to see that our worms, our normal worms, are able to lay about 250 to 300 eggs, which is expected. But our worms missing TANGO2 are not laying nearly as many eggs as we would expect, which again tells us that these worms are having some kind of metabolic problem. All right, so we then also wanted to look at a more sort of short-term look at metabolic processes. So something that requires a lot of energy for worms that they should be able to do pretty well is swimming. So if you put worms in liquid, they should be able to swim more or less continuously for about 90 minutes. And so when we observe our worms for periods of 4 minutes over a 20-minute period, and we score how well they're swimming, our normal N2 worms are able to swim very well, very rapidly, and more or less continuously for this 20-minute window. However, if we're looking at our worms that don't have TANGO2, these worms are swimming much slower, they're stopping, they're taking breaks, they're not able to maintain the swimming motion that they need to. And this difference starts even right away after they're in this liquid for even just a couple of minutes. And so this is a quantification of how much these worms are moving in this little 4-minute window. Here our normal N2 worms are able to sort of thrash their body to swim. And they do this very, very quickly for this period. However, our double knockouts are thrashing much less slowly, so they're just not they're not generating the motor capacity that they need to thrash as quickly as our normal worms. All right, and then one last behavioral difference that we noticed in our double knockouts. So under normal circumstances, worms live on these little dishes, and they eat bacteria, E. coli, yum, to survive. And so ordinarily, there's a little lawn of E. coli on the dish, and that's where their food is, and that's where they like to hang out, because why would they go anywhere else? All their food is right there. So normally, they all hang out sort of in the middle of the dish, and then, however, if their food source is bad, maybe there's a poison inside the food, the worms are going to eat this, their cells are going to get stressed out, and then they're going to move off of this food because they're like, wow, this is bad for me. I need to go look for a better food source. So they'll all hang out at different parts of the plate. However, previous people who have studied worms have also found that if you give worms that have metabolic problems, so their cell metabolism isn't working correctly, they're not generating enough energy, or they have lots of what are called reactive oxygen species, these worms will also move, even if their food source is totally fine, these worms will also move off of their food because their cells are stressed out, so they think that they're being poisoned, so they try to move away and find better food. And so we noticed that in these double knockouts, our normal worms in our lab are, as we expect, mostly hanging out within this little circle of their lawn. So they're eating their food, they're happy, they're having a good time. But with our double knockouts, our worms missing TANGO2, these worms are sort of scattered about our plate. They're about 50% of them are on our lawn at any given time. The other 50% are crawling off. Sometimes they leave the plate altogether, they go missing, and we can't find them, which that's never what you want. But so to us, that again also tells us that when our worms are missing these equivalents to TANGO2, they're having some kind of metabolic dysfunction that is telling them, you're not getting the nutrients that you need. You need to go look for them somewhere else. So having created this sort of profile of some of these differences that we're seeing in our worms that are missing TANGO2. Because we know that vitamin B5 has been helpful in patients and in other models, we wanted to know if B5 is also helpful for our worms. So going back and looking at some of the previous experiments that we did, so looking at these brood sizes, so this is the number of eggs that these worms are laying. We wanted to know if B5 could maybe help out with this. And so what we saw is that our worms that we treated with vitamin B5 are able to lay more eggs than our double knockouts, but they're not quite getting back up to the same level as our wild type worms. So when we're thinking about why might this be happening, this is helping a little bit, but why aren't they quite getting back to normal like we would expect? Going back to this slide, when this compound is split, like I said before, it gets turned back into B5, but it also generates this other compound called cysteamine that's important in a lot of other metabolic pathways that acts differently than TANGO2. So if our worms, like we think, have a problem at this step in this recycling pathway, even if you feed them a lot of extra B5, they're still not going to be generating this extra cysteamine. So maybe this compound is also important, and our worms are not getting it when we give them B5. So if we give the worms extra cysteamine to try to rescue them, we also see that the number of eggs that they lay gets a little bit better. So then our next question was, okay, if we give them both B5 and the cysteamine together, so these two products from that last step, is that able to maybe make them better? And no. So this was kind of unexpected. It was a little bit unexpected for us. This did not help them get better. They're not significantly worse, but this was not the improvement that we were hoping to see. And so we have a couple different hypotheses that we're working on as to why this could be happening. It could be that there's other things that the worms need when they're missing TANGO2. There's also just some technical things with administering these compounds to the worms. So we are supplementing them by, dosing their bacteria with these extra compounds. And we don't really know what giving these extra vitamins to the E. coli is doing to the bacteria itself. So it could be changing the amount of B5 or cysteamine that the E. coli are producing. So there's some other questions that we're working on exploring in this particular step. And then we also wanted to look at other aspects of worm behavior. So then circling back to the lawn avoidance that I was talking about earlier, we also tried to rescue this with B5, with cysteamine, and then with our combo of B5 and cysteamine, and we saw fairly similar results. So our worms without TANGO2 are hanging out off of the lawn. When we give them B5, they get a little bit better, more of them are on the lawn, but they're still not quite up to the level of our normal worms. When we give them cysteamine, that also helps them stay on the lawn a little bit more, but then giving them both B5 and cysteamine doesn't make that significantly better. So they're still not quite where we would want them to be. So when we were thinking about why this might happen, we had a couple of different questions. So one of them was just about if there could be something going on in the background of the worm strain that we were using, because this was a strain that our lab didn't personally generate. So while I was working on these, I was also working on generating my own double knockout strain. So I took some of our lab normal worms, went into their genome, and took out both of these two TANGO2 homologs. So these are worms generated in-house that are also double knockouts. And so went back and looked at if there were any differences between my double knockouts and the previous double knockouts that we had been working with. So we went back and we were looking at, went back and looked at our CoA phenotype, and we saw that similar to our previous strain, the double knockouts have a good amount of CoA at baseline, and then when they're starved, this CoA level drops off. There is a little bit in this experiment, the N2s also dropped a little bit, but I think this is due to some updates in how the CoA was being measured by our collaborator. And so current stuff that we're working on is also looking at our results in our homegrown double knockouts. And so when we're looking at some of our behavioral studies, we can see that the behavior of our worms is kind of in the middle of our normal worms and this other double knockout. So our brood size is variable and intermediate. Our worms also, their swimming is okay. So at baseline, they're actually able to swim about as well as normal N2 worms. However, if we stress them out, they do start to have a little bit of the previous defect that we saw in their swimming. So going forward, to try to address some of the future questions that we want to get to, we're looking at how we can look at our worms in liquid culture. So looking at them in liquid allows us to very precisely control the amount of nutrients that they're getting in their food. And it doesn't have any E. coli in it, so any of the questions about what these compounds do to E. coli are kind of taken out of the equation. So we can look at them very quickly. And we're in the process of optimizing this and getting pilot data that we're hoping to turn into a high throughput drug screen, which the TANGO2 Research Foundation has very generously funded a grant for some upcoming work in that. So stay tuned for some updates on that. And so in conclusion, my worms that are missing these TANGO2 homologs have this distinct phenotype. So they live shorter, they don't reproduce as much, they have impaired movement, and some lawn avoidance, which suggests that they have some kind of metabolic problem. And when we supplement them and try to rescue them with B5 and this other compound, cysteamine, we're able to partially rescue them, but not fully. So we have more work to do on that front. All right. So that's the end of my talk. I just want to thank everybody. So thanks so much to the TANGO2 Foundation for having me talk to you guys and for generously funding some of our research, our lab, and all of our wonderful collaborators. Yeah, this has been awesome. Thank you.